Great. Good morning, everyone. Welcome to this very special seminar organized by EvoFunPath. And I can tell you that ever since EvoFunPath was funded and started, it's been on Sarah, Professor Sarah Gurr has been on the top of our list as a speaker. So, and she's very busy. She's traveling the world. She's solicited everywhere. So we're glad to finally have a chance to welcome her in snowy Quebec City. She was able to extend. She was part of a very important meeting, the CFAR meetings, that like for the biggest or most important thinkers in fungal biology. They convene every year or so to discuss what is important. So obviously Sarah is part of that. Sarah's had a very, very important in terms of studying. She's from the UK, so she got her PhD. Correct me if I make a mistake, but at Imperial College of London. So if you look it up, you'll see it's one of the top universities. She got her PhD at the tender age of 23. Most of us are wondering what we're going to do next, and then she got tenured at the even more tender age of 28. Really anecdote, very, very important. You've all heard of Oxford University. So Oxford University was founded in 1409, so one of the oldest universities in the world. So 1409, and Sarah was the very first female faculty at Oxford to be integrated into the non-inclusive system of way back then. So that was quite a feat. So, and then obviously she made a mark at the time. That's where we met. Like a powdery mildew was the common point of interest. And then she was heavily solicited. And in 2014, I think, then she was offered the privilege of going to Exeter University to work on a very, very interesting and it's fascinating. We were talking about that yesterday, but the global movement of crop pathogens, mostly with fungi. So the EvoFunPath is obviously focused on fungi. So, and then she's been involved in this field and she's been extremely successful. I think she has over 14 or 15 papers in Nature on this subject alone. And then she's known really as the world expert on all this aspect of how pathogens spread and we create threats. So that's why this morning we are very lucky to catch her in between two flights. So she'll give a talk, and after that we'll need to rush to the airport. So I won't take more of her time. I'll let her talk to us about global movement of crop pathogens.
So thank you, Richard. That was a very kind introduction. Makes me blush. Good morning, everyone. So I'm going to talk about the global movement of crop pathogens, perils and predictions. So the outline to the talk is I'll start with some global challenges, then I'll turn to some of the modeling we've been doing, and I'll try and go at two levels. I'll go at the global level, and then I'll go down to an example of a particular pathogen at field scale level. I'll make some brief comments on fungal emergence and adaptation, and some brief comments also on the need for new fungicides and stewardship of those fungicides and discovery, and then a few comments about the way ahead. So let's start with global challenges and talk about food insecurity and pests and pathogens, and most notably fungi and oomycetes. So I hardly need to rehearse the fact that we have all sorts of challenges to global food security at the moment, not least the expanding global population, with, I understand, it reaching 8 billion last week or so, and projected to rise to about 10 billion over the next 40 years. So that's a very significant number of mouths to feed. We've got unbelievably volatile political conditions through the world, such as conflict and war and migration. We've got spiraling energy costs, particularly over Europe. We've got land degradation through the world, whether it's by urbanization or soil erosion, and that's a particular problem in China. We've got water crises such as droughts and deluges, and we've got dramatic weather as well as temperature rises. But add to this pot, we've also got pests and pathogens. And of all the pests and pathogens, the most important, which I'll show on the next slide, are the fungi and oomycetes in terms of crop disease. And that's rather different from the balance of human disease, where viruses and bacteria are more important. The sobering fact is that despite mitigation, so despite spraying fungicides and breeding for disease resistance, we still lose between 10 and 23% of our crops each year the fungi and oomycetes. So this is making it rather quantitative. So along the axis, you've got the passing of the decades and the records of the different pests and pathogens. And I just want to highlight two things on this, which is the yellow, which are the fungi, and the oomycetes, which are not insignificant in number. So the most challenging pests and pathogens are the fungi and the oomycetes in terms of food insecurity. So now I want to turn to look at two things, which is the calorie crops and the commodity crops of the world. So if you look at wheat and rice and maize, they cover 40% of global agricultural land. The pathogens that wipe out these crops are fungi, such as wheat stem rust, rice blast, corn smut, and then we've got soybean rust, and also we've got Phytophthora, an honorary fungus, the oomycete, Phytophthora infestans, which causes potato late blight, the cause of the Irish potato famine. Despite mitigation and not during epidemic years, we still lose to these crops alone enough to feed between 600 and 4,000 million people, 2,000 kilocalories a day for a year. So we're losing a lot of our crops, even though we're trying to protect them. And what is important also is to realize that whilst this pathogen on the left is an important rust fungus, a rust fungus of tropical crops in temperate lands, particularly in Europe, our major pathogen on wheat is this fungus. It's called Septoria triticide blotch. The fungus is Zymoseptoria, where we've labeled up with GFP the zymo cells. And I'm going to talk about this fungus later. So I look to see whether it's important in Canada. And I see that you have 3 rust fungi, and this one is not yet important in the wheat fields of Canada, but maybe it will come. And I also wanted to highlight the fact that whilst we have these commodity crops which directly feed the peoples of the world, calorie crops, we also have commodity crops. And some countries are completely reliant upon the export of their commodities to allow them to then buy in the calorie crops that they need. So for example, Colombia has an economy which runs quite heavily on coffee rust, on coffee, and this is the fungus which periodically wipes out their coffee harvest, coffee rust. And you're probably aware at the moment that there's a new threat to bananas with tropical race 4 of Fusarium rushing through, particularly Cavendish cultivar bananas. And these bananas, Cavendish, form 90% of all export bananas in the world. So the point really is to introduce the fact that fungi challenge food security by challenging both our calorie and our commodity crops, and also in post-harvest storage diseases, which I'm not going to talk about. So I want to turn now to ask some basic questions and to try and show you how we build our global and our field scale models. So this really started with an expedition to the pub with a postdoc of mine called Dan Bebber, who's now got a professorship in his own right. And we just started messing around thinking, what sort of really basic questions can we ask about crop pests and pathogens? So where are they? Are they on the move? And do they move in concert with climate change? Which of these fungi and oomycetes are going to pose the greatest threats? And when and where will they occur? And so we began to think, what sort of models could we run? Could we run predictive distribution models? And could we also model across the scales of sizes at global, field, crop and pathogen level? And then also eventually, what can we do to prevent disease? So we've taken 2 approaches to crop modeling. Up from 2013 to about 2018, I think what I like to call them now are the naive models. They are statistical models. And from more recent models, we've gone mechanistic. And what I want to just highlight here is the fact that the mechanistic models are powered by data that we've begun to gather in the lab. So let's start with the basic questions, and this is Dan. So what is the global distribution of pests and pathogens? Are they on the move? And can we predict the greatest threats? And when and where will they occur? So let's just think more about geography at the moment than pest biology. So what determines the global distribution of crop pests and pathogens? Well, if you think about it, the most obvious thing is that the number of pathogens should reflect the volume and also the diversity of agriculture in a given country. We can then conject that there might be more pests at the equator. We can conject that because that will go with species richness. So where you see a latitudinal gradient in species richness, as in wild species. And then the third conjecture is that, according to island biogeography, it suggests that lower immigration rates and higher extinction rates are found on islands. We can conject we find pests on islands. So it's all very well to pose these questions, but how do you begin to answer them? So these are our naive models. They're generalized linear models run in an iterative process. But what sort of data could we get hold of? Well, in the 1890s, the Commonwealth Agricultural Bureau, which has now been renamed CABI for all sorts of reasons, began to collect records from around the world. And in 1960, these data records became robust, meaning that a single observation wasn't good enough. You had to have 3 observations of a particular pest or pathogen for it to be valid. And so an undergrad project student took 85,000 pieces of paper and digitized it for us, poor chap, and he looked at 2,000 pests in all the countries of the world. We then went to the Food and Agricultural Database, and we wrote down the 168 crops, which are important calorie crops. But what you can't do is, if you do this sort of modeling, is you can't say, let's compare Canada and America with Haiti and Myanmar. So to try and level up the countries statistically, we took into account the gross domestic product per capita in each country, as well as how much a country was able to spend investing itself in research and development. Then we got all the agricultural data for imports and exports. And we're lucky in Exeter, we have the Meteorological Office. We also have access to the US satellite data and also the Japanese weather data from around the world. And then we classified every country in the world as to whether it's an island, whether it's landlocked or coastal. And we got 2 things out of this. Firstly, the current distribution of pests and pathogens in the world, and then a model that allows us to predict pest and pathogen presence. So the first stunning thing, which was completely what we did not expect, is this, where you're looking along the bottom at per capita GDP, and then you're looking at pests per ton of agricultural production up the y-axis. Each country here is either a closed circle, landlocked is open triangle, or coastal is a square. And what you can clearly see is this extraordinary thing, which there are more pests on islands than there are in landlocked regions. So we asked why. So 2 countries in the world which have been really hot on biosecurity are Australia and New Zealand, and they're over at this corner of the graph. What's going on there? And the answer is, unfortunately, they've done a lot, but it's too late. And so they've already imported a huge number of foreign pests and pathogens. And despite their very robust biosecurity at the moment, things are still arriving on soil, in postage, parcels, and also on people's jumpers as they get off aeroplanes. So all sorts of problems have happened, but this was surprising. And then people have been more interested in the predictive model. So what you're looking at here is obviously a map of the world, and the green means that countries are over predicting, and the red axis is countries are under predicting. So let's compare America, green, with Myanmar over here, a really poor country where people are living on less than $1 a day. So if you equate this or equilibrate it, the data by taking into GDP, what you can see at the moment is Myanmar is currently investing very little money in research and development, and it's reporting about 370 pests and pathogens. But if you then take the model and you run it against a model from America with high GDP and research and development activity, it should report 67 or 670 pests and pathogens. Now, 2 countries have taken this data really seriously. The first is India, and they've actually changed their policy to go out looking for the missing pests and pathogens, and they've found them. And Madagascar have also taken it seriously because their economy is running on rice at the moment, and they're going to shift their economy from rice to yams in the face of disease and trying to get rid of so much starvation in that country. So are they moving? And again, we can go through all sorts of different databases, which I don't want to go through again, and always correcting for observational bias by taking into account GDP and how wealthy a country is. So this is the summary of all of the data of all the different pests and pathogens. And I want to draw your attention to the right-hand graph, which is the fungi only. So you're looking at the passing of the decades on the x-axis and the distance they're moving from the equator in red along the middle. And you can see as the decades are passing, fungi are actually moving, and they're moving polewards in a warming world. Oh, I do. Thank you. Wonderful. Okay, so here you can see with the equator, they're moving, and they're moving faster and faster. And you can see this is in concert with climate change, which I'm not showing here, but they're moving at a staggering almost 8 kilometers per year. So this is all very well. We can look at the distribution and we can predict they're on the move. But which of these fungi and oomycetes are moving fastest? And when will they arrive in different countries? So again, we went back to the databases, and without going through what we did, we actually took the national and subnational levels of every country. And for the first time ever, we assessed our data by matching every pathogen in our database to its host, having an index of host specificity. And this is important for one of the models I want to come back to later. So we then compared countries around the world by ordination analysis. And this is what we saw. So this is actually more optimistic. So all the regions of the world are divided according to the South Americas, Northern Americas, Africa, Eurasia, etc. What you can see about all the crop pests and pathogens is they're still strongly regionalized. So despite the current problems with climate change, our pathogens are still regionalized. Climate change will affect the distribution of this and the mixing, but we've still got time for really robust biosecurity if we take these models seriously and we prevent the movement of pests and pathogens ourselves by trade and transport more effectively. So what are the greatest threats of all of the fungi? So here the fungi are more threatening, obviously, than the other pathogens, and they're saturating the crops fastest. But which are going to pose the greatest problems? Now, this is exactly contrary to what we thought. We thought that the generalist pathogens would be the most threatening in terms of climate change. So things like botrytis, grey mold, that you find in the supermarket, a fungus that is necrotrophic and infects over 180 different genera of plants. But that's not what we found. We actually found that the threats are going to be from the mildews and from the rust fungi. So for example, the rust fungi and the diversity of rust in the Canadian fields is likely to increase as more exotic rusts come in. Now, please hold in mind this comment specialist fungi, because it infected what we did in terms of one of the modeling programs I'm going to show you. So these now we consider naive models. They took large data sets and they are simple statistical tests to show what was going on. But we decided we could be cleverer and we could use mechanistic models. So what we did is we took a very old concept, and the concept is called the disease triangle. And it's only a concept because it's never been parameterized. So what it says is disease is abundant in the fields if the host is susceptible, in other words, it doesn't carry resistance genes, if the pathogen is abundant and virulent, and if the environment predisposes the crop to disease, so in ideal conditions for the growth of the pathogen. And then we thought, well, we can be a bit cleverer, which is why we've got the Dali cheese here, the clock. We can make this three-cornered triangle into a four-cornered triangle, if such a thing exists, by putting a temporal axis onto it. And we did it really starting very simply. And we started looking at temperature to parameterize this disease triangle. And just to remind you, this is so basic, but it really has helped. Looking at cardinal temperatures by measuring the thermal limits of various biological processes in fungi. So we're looking at CT optimum with CT minimum and CT max. So we're looking to see whether this can empower this model. And we were stunningly lucky for two reasons. One is we had a superb PhD student, Tom Chaloner, who not only gathered the data for two of these fungi ourselves in the lab for the organism that causes septoria triticide blotch on wheat, and this one, Magnaportha, which causes rice blast disease. But the second lucky thing was in an antiquarian bookshop in Paris, I found this book, and it's written by a Japanese chap who spent his whole life and published in 1949 looking at the temperature of all the pests or the fungal pathogens and oomycetes that he could. The problem was almost all of them had changed their names. So Tom abstracted from his book and from his own data the growth of TT, temperature optimum, temperature minimum and max for a whole raft of different ascomycete, basidiomycete and oomycetes. In fact, 631 fungi and 64 oomycetes. And he characterized them according to growth in xenic culture. Obviously, you can't do this with biotrophic fungi. Their germination temperature optima, infection, and disease. And he's been able to do two things with this data. The first is we took our disease triangle ideas and we married it up with niche theory, which is ecological niche theory. And so what we were able to do then is to ask a question: if you're a true specialist or a true generalist in plant pathology, does that define your temperature optimum? And we found by evaluating the cardinal temperatures for all of those life cycle processes in fungi and oomycetes and matching them all to their hosts assessed by phylogenetic diversity, we found this, that although they are generalists in terms of their host preferences, they don't have a wider temperature range than specialist fungi. And in retrospect, it probably seems quite obvious, but it does rather redefine the notion of what is a specialist and what is a generalist if you look at ecological niche theory with plant pathology. Unfortunately, that gives us a rather gloomy outlook because it shows the remarkable adaptability of all fungi and oomycetes to new climates and new hosts. What I wanted to return to is just this box here, which is the infection data, because this was far more exciting in terms of what we found out from it. So just to remind you, we're now beginning to build a mechanistic model to predict global pathogen burden in the future by parameterizing, first of all, this corner of the three-cornered triangle. So we've got the pathogen data that I've showed you, which Tom had abstracted both from the book and gathered himself. But we can overlay that with host data. We can use three different yield models that exist, and we can also match both temperature and humidity to these host models. And then we can match this to host range using different other models. And then to get the third corner of this, we can use different climate models according to the International Panel of Climate Change. And we've chosen this 6 value because this is probably the one that we're most nearly covering at the moment in terms of global climate change. So all of this gives us absolutely huge amount of data, not only about all the crop species, but also about all the pathogen species as well. So we did 2 things. We ran the models simply by looking at the crop alone, and we put in calories, soil, all sorts of stuff, all the crops. And the first model we ran, we had to get a supercomputer at the Met Office to do it for us. We knew our model was completely wrong because it told us we should be growing coconuts in northern Russia. So we went back and we parameterized all of the host data and then began to match it with what was quite sensible. So we had that, and then we could overlay onto that model all the pathogen data. So it's all very well modeling, but it's no good if you are unable to compare it with real data. So we were able then to go back to validate the models by comparing this with the original observed pathogen presence from the PABI databases. And we were able to level it out through the world with GDP and research output, and we were able to do it at the regional scale. And that allows us to project forwards in terms of the movement of crop pests and pathogens to 2070. So what we're looking at on this map is the changing global burden of now compared with 2070. So the global burden means you have less pathogens if you're blue and increasing pathogen levels if it's red. And these are the different months of the year. So if you look at Canada, you can see that you're going to get peaks in infection in late autumn and also early in spring. Now, this has led to quite a lot of interest in this paper, particularly with countries who say, well, we're already growing a particular crop. Should we be growing a different crop? And how long will it be before the pathogens arrive? So the main message from this is 3 things, which is that the crop models reveal yields will increase in higher latitudes but decline in the tropics. That means there'll be less food around the Sahara, around the equator, which is leading to greater food inequality. And some regions, such as America, Canada, Europe, and China, will then see very different changes in the pathogens that arrive. However, over time, we'll go back pretty much to the status quo because there'll be a honeymoon period when the new crops are built or are grown in different latitudes. And then, unfortunately, the honeymoon will finish as the burden of disease will either be from known pathogens or from unfamiliar pathogens on naive hosts. So what we've been able to do is to parameterize the disease triangle and make it from a concept into a paradigm. We've done this at the global scale, and we've intercalated temporal data. But we can also do it at the field scale. And I'll only very quickly show you how we've done this with this pathogen. And I've introduced this before. This is Zymoseptoria, which causes Septoria triticide blotch. And it's the most devastating fungal disease of wheat in Europe. And I'm truly European, so I've always said UK is still in Europe, but it isn't, unfortunately. And to parameterize this, you need to know your enemy. So you need to know a lot about the pathogen. So Tom in his PhD labeled up this pathogen with green fluorescent protein, or M-cherry, and he followed the life cycle of the pathogen by confocal microscopy, counting what happens on the leaf surface at different temperatures and different humidities. So he was able to start to build a model to say, how do we now feed this into the disease triangle? So the data he collected were temperature response functions for germination, growth, death, and for humidity. And he was also able, from spore trap analysis, to look at the spore loads out in the fields. We were able to get maps of the UK to get the spatial resolution of UK wheat. And then we've got these very high resolution data sets from the Met Office, defining the field infection periods by temperature, humidities. And we were able to validate this model by looking at what's happening in organic farms through Britain. So we're able to get this data. So we've got the first ever predictive epidemiological model empowered by the disease triangle, but it has all sorts of limitations because it's still very naive. But what did it look like compared with anyone else's model? Now, this is the best model that exists. It comes out of a French group, Bernard et al. And what they've done here is they've tried to take into account some of the basic biology of the pathogen. But as you can see, when you look at the predicted infection risk versus the observed one, their correlation isn't very good. But ours isn't bad, but it's still not good enough. It's 0.61. So we can say where in the wheat fields of the UK we predict this pathogen will occur. So how do we improve this model? Well, we've informed it with pathogen biology, environmental parameters, and some very basic data on the host. But we can do better, particularly with the host and particularly with the pathogen. So with the pathogen, we can use field isolate data, which we're beginning to do with this particular pathogen. We can use host resistance data, the presence or absence of our genes, and we can use fungicide treatment data. And that's what we're doing at the moment. And other groups have asked if we can join up with them to try and predict the movement of their particular favorite pathogen. So just to finish off this, and the next part of the talk will be far briefer, what we've done is we've gone across the scales of sizes. We've taken global models, field scale models, and now we're beginning to look at cellular responses, particularly the impact of temperature and humidity on pushing fast forward evolution in the pathogen itself. And this resonates very much with the global burden of crop loss initiative, which is funded by CABI in association with the Bill and Melinda Gates Foundation. I have money from CFAR, and it also resonates with the Foreign and Commonwealth Office of the UK. So let's just think about what's going on in the field. So why are we in such jeopardy in terms of the emergence of new fungal diseases? Well, we've got double jeopardy because we've got global changes such as climate change, and we've also got problems with trade and transport. But probably the biggest force forwards is the modern agriculture practices, because we've got new variants of existing pathogens coming across our fields. So just out of curiosity, I looked in the world to see where the largest wheat field is, and it's here in Canada. It's 14,168 hectares of wheat. Now, this is a monoculture. And this is depressing because this is a single cultivar of wheat, which is guarded by one resistance gene, and it's sprayed perhaps 7 times during the season with single target site antifungals such as strobilurins, azoles, or succinate dehydrogenase inhibitors. What a feeding and breeding ground for the pathogen. So let's just look at this quantitatively. So we've got these vast feeding fields of genetically uniform, poorly guarded crops. The pathogen's having a ball. It goes through many, many reproduction cycles during the season by producing these pycnidia on the leaf surface. So here we've labeled one up with GFP and we've counted the spores. So in a single season, in a hectare of a wheat field, we can estimate that there are 100,000 million, so 10 to the power 11 spores. Now, from the genome sequence of this fungus, it's got every architectural hallmark you could think of for fast evolution. So it's got retrotransposons, effectors, SNPs, and everything. And it's literally emerging new races of this devastating fungus frequently. And this is the same as happening in Magnaportha, so the rice blast pathogen, and all the other major pathogens. So monoculture is great in terms of yield, but it's also great for the pathogen. So what we're effectively doing is taking this concept of the disease triangle, and the hand of man is making it from an equilateral into a scalene triangle, because we're pushing the fight, and the fight is the war of the kingdoms, not really now between the plant and the pathogen, but between us and the pathogen as it fast evolves. So what are we going to do about it? And at the moment, there's some brilliant projects around the world using gene editing, but we have to be very wary that if we edit our genes, they might also be ephemeral and the pathogen can evolve to overcome them. So we have to be more resourceful in the way that we're genetically modifying crops for disease resistance. At the moment, fungicides remain our best weapon, but there are problems with fungicides. So about 53% of the fungicide market is dominated by the azoles, which are ergosterol membrane inhibitors, by the strobilurins and the succinate dehydrogenase inhibitors. These fungicides target mitochondrial respiration in the fungi. They are all single target site antifungals, and of course the pathogen rapidly has overcome these to become resistant. So they are ephemeral. You put a new azole out in the field and its lifetime before resistance occurs is 2 years. The same with the strobilurins and the same with the succinate dehydrogenase inhibitors. So what we need to do is to be wary of this and really go to mixtures to mitigate risk of resistance emergence, and we desperately need new antifungals. So we looked at the worldwide emergence of resistance to antifungal drugs challenging human health and food security. So here are the crops, and here are animals and humans. And I just want to highlight the azoles. The azoles remain the frontline weapon for the clinician if somebody reports wheezing or whatever. And after COVID, many people had systemic or lungs filled with Aspergillus fumigatus. When you take these Aspergillus fumigatus strains, they are almost invariably azole resistant. So there's been a bit of an outcry from the clinicians saying, we can't use azoles in our crops, we must save them for human patients. And then us saying, well, if you don't have azoles, you won't have anything to eat, and so you probably won't get COVID anyway. You will have died of malnutrition. So that's quite a political fight going on at the moment. So in terms of the movement of azole resistance over the last 40 years, this is a really simplistic map. This is just simply taking 40 years of data of 6 human pathogens and 52 crop pathogens and looking at the movement of azole resistance around the world. And it is quite sobering. So this is just pure data extrapolated by a brilliant student, Nicola Hawkins. So what are we going to do? We desperately need to protect our crops from disease. We need to take the discovery of antifungals away from single target sites to get multi-target site fungicides. And we need to take the profit out of it so that everyone in the world has equal access to chemistry to control disease if we know that the chemistry is relatively benign in terms of sustainability and environmental toxicity. Well, my colleague, Gurr Steinberg is a very clever fungal cell biologist. And what we decided to do was to see if we could take any lead chemistries and invent a new fungicide. And there are fungicides on the market, such as a chemistry called dodine. And dodine is used in Canada and in Australia and New Zealand to prevent venturia, which is apple scab. So we took dodine, and with a brilliant organic chemist called Mark Wood, we asked him to make it so that the molecule had a very long lipophilic tail, and we wanted to put a sulfonium, so a sulfur head, into the top of it. So we effectively have something that's just like a detergent. And we managed to show that this lipophilic cation, which has many modes of action, it targets mitochondrial respiration, it targets reactive oxygen species, and it also seems to boost the innate immune system in crops. So we're quite excited by this. And at the moment, this has just been recently taken up by a small company in Germany, and hopefully it won't be a profit fungicide if we can control it. And it's also working at the moment to suppress the very disease, Panama disease, which I mentioned as one of the important commodity crops at the earlier part of the talk. So here, this is Panama disease caused by tropical race 4 of Fusarium, and this is a control plant. And what we've managed to show using this fungicide and all the other fungicides, so we took 7 different classes of fungicides and we looked at their efficacy on different spore forms. And here we developed a healthy plant index, and we used healthy plant index and corm necrosis to show that of the various chemistries, ours, which is C18-DMS, and dodine, which is the known fungicide, are suppressing this disease about 80%. And when you look at it, you can see that this will probably go on to produce bananas. So we haven't got a cure, but we've got disease suppression, and it's a multi-target site fungicide. So I'm going to finish now by asking, what do we need to do to meet these upcoming challenges? Well, the scientific requirements are that we must understand the pathogen. It's pathogen, pathogen, pathogen. And unfortunately, there are hundreds of different pathogens and hundreds of different hosts. We need to be able to model the movement of the pests and pathogens. We need new antifungals. We need to understand why fungi become resistant not only to the antifungals, but to the R genes. And none of this research is possible if we don't do it in an interdisciplinary way. And we're constantly needing new tools and techniques to investigate these sorts of diseases. And our societal requirements are that we need to raise public and political awareness, particularly political awareness, as to the plight of plants to fungi. And we need to be able to influence policy to ensure that we have better biosecurity. And we need education and succession. And that's why you are also important at the University of Laval to lead this sort of area in Canada. So with no more ado, these are my lab members. Dan, who's now made it to full professor in his own right. Tom, who was such a gifted PhD student, ended up with 5 papers in Nature journals. Helen, my postdoc, and Will, my postdoc, who's done all the banana work and all the funding that I've described, and all my collaborators, most notably Gurr Steinberg. Thank you very much.